why is it important to study ASD in Qatar? And so if you look at large global efforts around ASD, you'll see these major efforts: Autism Speaks, Agri, Spark, Safari. They all have thousands to tens of thousands of patients that they've already studied. And so through those studies, there's over 1,100 genes that have been found associated to ASD worldwide. And so there's many questions that come to mind. Are these the same genes in our population? Are they going to be different? And how do we know? And so if you look at the Middle East, in comparison, there's very few studies. They're mostly fragmented studies. We know that in Qatar we have about 6,000 to 8,000 families that have at least one member with ASD. A recent prevalence studies shows that it's around 1 in 88 children have some form of autism or are on the spectrum in the country. And to be honest, when we looked at the literature back then, we only found a very small number of genes, maybe around 20, that had been identified in the GCC in families with autism. And so main questions that come to mind are these genes specific to our part of the world, or do we share any genetic architecture with global populations? And if so, how can the knowledge of genes help us with personalized treatment and intervention? And that really was the motivation for the BARAKA study. You may have heard of the BARAKA study, but the word baraka was selected for its dual connotation. So in Arabic, we all know that the word baraka means blessing. And that really is what we're studying when we study patients with autism. At the end of the day, every one of our children is a blessing from Allah. And then BARAKA is also an acronym. It stands for Building a National Resource for the Advancement of Knowledge of Autism in Qatar. And so I'll talk a little bit about how we set up this study and some of the preliminary findings that we have. So the very high level, what we do is when patients want to enroll, we usually schedule an appointment for them. They come to the hospital, they consent for being enrolled in a research study, which is very different than a clinical care approach. With the research, we do a lot of different questionnaires that we administer to try to look at some of the environmental impact of autism. All of our questionnaires are benchmarked in collaboration with Sick Kids Hospital, which is a global leader in ASD research. We then collect and biobank samples from the patient, from the family members, from siblings, whether they're healthy or affected. We really want to try to build a big resource for autism. And then we usually do whole genome sequencing on these individuals. So we've actually started multiple enrollment sites. We enroll at Sidra from multiple different clinics. We enroll at HMC. We also have collaborations with multiple centers inside the country. And as I said before, we collaborate with Sick Kids. And through Sick Kids, the entirety of our study is actually represented on the Autism Speaks Google Missing platform. So if you go to Missing, you'll see that it's true that they have 100,000, they have tens of thousands of patients now from around the world. And then you'll see a small cohort of 100 from Qatar. And really our goal is to grow that cohort as much as possible to give the Middle East larger representation there. So it starts by identifying families. This is usually done in the clinic. And then once the families are identified, we send them all for routine pathology. And so we rule out any families with known causes. So if the child has Fragile X or has a good microarray finding, those would usually not be enrolled for further research because the genetics are known. However, for all the other families, we enroll them into this research with very wide consent and a very detailed environmental questionnaire. And then, as I said, we do a biobank of all of their samples. The first pilot phase that we just published was on around 100 families. So this was 372 participants because when we count the number of children with autism plus their parents and their siblings, the majority of them are simplex. Simplex means there's only one child that's affected. And then we had 2 families that had multiple kids who were affected with autism. As you'll see, the age, this is mostly going to be pediatric because Sidra Medicine is a pediatric center, but we don't have any restriction above 18. And as we know from autism, the male to female ratio is 5 to 1, and this is consistent with global averages. So we also know that autism has many comorbidities. This is just a different way of looking at some of the comorbidities. I have them in tabular form here in the left. In the majority of our patients also have ADHD. Some have intellectual disabilities, some have speech delay, and some have other comorbidities. This on the right is just a different way of plotting this for a scientific audience, where we show the frequency of the different combinations of comorbidities in our cohort. We also look at the study demographics. The study is open to literally anyone. We do not have any selection criteria in terms of ethnicity or in terms of country of origin. Any patient with ASD can be enrolled. The vast majority, by chance, happen to, in the first 100, happen to be of Arab and South Asian ancestry. And as you'll see, there's relatively high levels of consanguinity. Consanguinity means that parents are usually related, and this is not surprising for our part of the world. We do see this not just in ASD, but across many different rare and undiagnosed disorders, etc. So as I said, over 85% of our families so far have been in MENA and South Asian population. So this is a one-shot look at everything that we do from a whole genome perspective. I don't expect anyone in the audience to fully understand this. It's a fairly technical slide. But the point I'm trying to make is that when we do whole genome sequencing, we are really doing comprehensive analysis of every single thing in your genome. And this is really state of the art. Most labs that send a sample for genetic testing will look at one or a few genes, or they might look at one or a few types of mutations. But really the pipelines that we've built at Sidra over the last decade for genomic analysis have come in very handy when we look at autism, because autism is a very complex phenotype to study genetically. These are just some examples of families. There's families where you have, a single proband, and so we discovered mutation in this gene called STAG1. This gene is sitting in global databases, and it's known to cause syndromic ASD. It's also known to cause severe ASD with mental retardation. And as we know from our family, we do have intellectual disability and ASD present in this proband. This is a way of looking at it, at the genetic data also. You see that the mother and the father and the sibling, they don't have this red dot, and this red dot is really the mutation that's observed only so the proband really means the patient with the condition. And so there are articles on this STAG1 gene being involved in ASD. Here's another family where we actually have a father and 3 children who are all affected. And it turns out by looking at their genetics that they share this mutation in a gene called dynamin-1. And this gene is known to cause developmental delay and epileptic encephalopathy. And so we know a little bit about how this gene functions. And again, we see it shared by the proband, the father, and the 2 siblings, but not by the mother. And so this has also been published. We also see different types of mutations. So you might have heard of chromosomal microarrays. So chromosomal microarrays look at events, deletions, duplications, changes in the chromosome that are on the size of about 1 million bases or more. That's usually the resolution that you can look at with chromosomal microarrays. However, there's a lot of similar deletions and duplications that happen on a very small level. And here's a family where this child that has a very, very small deletion. It's only 2,000 bases. So it's only 2,000 letters out of billions, but only 2,000 have been deleted. And as you can see, the mother has only 1 copy of this deletion. The father has 1 copy, but this child happens to be homozygous. So you see this when you have consanguinity, which means the parents are related. That's how they both share this mutation in the first place. If they were not related, it would be very unlikely that they would have this mutation together. And then because they're related by chance, 1 in 4 of their children will be homozygous. And this is actually what we see in the proband. And so we see this deletion in EVOL2. And again, this is sitting in the Safari candidate with a very high score. In the interest of time, I'm just going to summarize our results real quick. So in 27 out of 100 families, we discover a genetic cause. So this is about 1 in 4 children have a clear genetic cause for autism. And most of the individuals that we had recessive autism. I'll show you this in a comparative shot. So on the right is the BARAKA study. This is our study. The red are the patients that we did not diagnose. So 73%, 27% we found a genetic diagnosis in them. When you compare this to big cohorts like missing and Simon simplex cohort, you see that we have about a twofold higher increase in establishing a genetic diagnosis. And we think this is because of this slice of the pie. So in global databases, no one really has recessive autism. It's very rare for parents to be related in global databases. Whereas for us, we have a large percentage of our pie can be explained by recessive mutations. And so this is statistically significant. This is just a way of showing it scientifically, that we have a significant enrichment for recessive ASD in the Qatari cohort. And what this means is that recessive conditions are easier to predict and screen for, so we can screen for it premaritally or even in a newborn, and this can support with prevention and intervention. This paper has been published at the end of last year. As you can see, many people, including Dr. Madiha on this webinar, have contributed to the study, and we aim to publish more. So I just want to go back really quickly in the last few minutes to this idea of having a biobank and why it's important to have a biobank for ASD. So this is not a one and done. This is not something where we study it once and then that's it. If you're negative, and if you're positive, you're positive, and then we give back results. This is really about thinking long term, about how, if we grow the patient population cohort, what we can actually do for the ASD community. And so this biobank is really important because from these same samples that we collected at research consent, we can do a lot of other work. Metabolomics and proteomics is another type of work that we can do in autism. So metabolomics and proteomics are just fancy words of saying that we can look at all other small molecules that are not DNA, they're not genes, but they're small molecules that are circulating in your blood, and they can tell us about your biological state. So we can look at thousands of these all together in a very, very small 10 microliters of samples. You cannot even imagine how small it is. So 10 microliter is 1/100th of a milliliter. So when you take 5 ml of medication, think of 1/5 of that and then 100th of that. So that's how much blood we need to be able to get thousands of proteins and metabolites in the lab. And so this is one study where we looked at the metabolomics. We took from the BARAKA cohort, because we have control siblings. That's why it's very important if you enroll your children to include both affected and unaffected, because they can really, really help with these types of studies. So we include cases and controls, and we look at, again, thousands of metabolites across their blood samples, and we discover metabolites that are what we call significantly dysregulated in individuals with autism. And as expected, these metabolites fall in pathways such as urea cycle, glutamate metabolism, arginine and proline metabolism, which are already known to be heavily involved in autism spectrum disorder. So it's good that we can find the things we expect to find in our blood samples. That means that our collection and our scientific approach is very sound. And this also means that as we grow our cohort, we're going to start finding new things that no one else has found that are going to be specific to our cohort here in this part of the world. We also looked at the proteomics. So we combined our BARAKA cohort with 2 other cohorts globally to become a large data set. And again, this work has found multiple protein modifications that are significantly enriched. So wherever you find a p-value that's less than 0.0001, you know that enrichment is highly significant in ASD versus typical development children. So these are normal development children. And so this has been replicated across 3 cohorts, and this is why it's important to create a biobank, because we can start contributing to global studies. And this has been replicated actually because our data set starts in very, very young age. We found that signal in the youngest age category. And this has also been published. This was work that was led by Qatar University. And again, this was because we had a biobank here and they applied for access. We gave them access to all the sample, and they were able to actually run this project themselves completely independently. So this is why when I say that the BARAKA-Qatar study is really a groundbreaking achievement for this part of the world, is because we are really building a national resource. This is not just for Sidra to use. This is really for the scientific community to use. So I'll end there, and I'll just summarize with a few slides. One is that we all know that ASD is incredibly multifactorial, so it has contribution from genetics and from environment. Using state-of-the-art whole genome sequencing, we discovered genetic cause in about 1 in 4 children, which you might think is low, but it's still double as high as international averages. So that's actually relatively high right now. Many of what we discovered inside Qatar has been recessive. And again, this is not Qatari only. This is all the patients that we had. And so when you find recessive mutations, it helps with screening programs in the future. The benefit of biobanking and why we ask participants to biobank, it might not be obvious at the start, but it's really because these same samples can continue to give and give and give to the research community in ways that you might not imagine possible today. As technologies develop, there's going to be a lot of discoveries that will be made in the future, thanks to the biobanking that we do. And again, if we really want to have the right amount of power to discover new genes and new biomarkers and new treatments, we need to grow our cohort. And we're now looking at pan-GCC efforts to try to get more patients from across the board enrolled in studies like this. So again, I just want to remind the audience that the BARAKA is a growing national resource. If you want to contribute to it because you or a family member has autism, please feel free to enroll. If you want to access it as a researcher, please feel free to access it. And if you're a young student sitting in the audience that's thinking about an undergraduate project or a master's or a PhD project, and you're not really sure where you want to start, but you want to do it in autism, please get in touch. We would be happy to help you and to guide you and for you to use this data set. So I will stop there. Thank you very much.